All right, so today we're going to finish up with our discussion of the endocrine system and start talking about muscles. We'll talk about muscles for the rest of this unit, and this is realistically going to spill over into our exam 4. So wherever we get to by our review is where we're going to stop on muscles. But let's pick up thinking about absorptive and post-absorptive states. This is on.
Finish up with our
Great. All right. Okay, so take a couple seconds just to briefly summarize what insulin does in the body before we start talking about its antagonist hormone, glucagon. Anyone want to share like one sentence about insulin in the body? Yeah, exactly. So insulin is going to be promoting glucose storage. Insulin is going to react during the absorptive state. It's our hormone of the absorptive state that's involved with storage, anabolism, when we have plenty of glucose. So we're contrasting insulin and the absorptive state with glucagon, counterpart during the post-absorptive state. So insulin was coming from the beta cells in our pancreas. Glucagon is coming from alpha cells in the same location. So in the pancreas, in these islets of Langerhans, these endocrine islands in the pancreas. Its job is to promote glucose sparing. That's because the brain and nervous system really need that glucose in order to do metabolism. So all of glucagon's actions are really going to be related to that role. So they're going to be, your body under the influence of glucagon is going to be mobilizing energy that it's stored, which means we're going to start breaking apart glycogen, so glycogenolysis. We're going to be breaking up our triglycerides that we've stored in our adipose cells. That's the lipolysis, or breaking apart our fats for energy. We're also going to synthesize new glucose. So because the body doesn't have this glucose coming in, and your nervous system needs glucose, we're going to take all the pieces we can find and do gluconeogenesis, make some new glucose that we can feed into the nervous system. So we think of glucagon as a catabolic hormone, so a hormone that breaks things down, because we're breaking down these storage molecules. It's the antagonist of insulin, which basically means that it's doing all the opposite of the things that we had insulin doing. So insulin was building things up, storing it. Glucagon is breaking stuff down, making sure that we save all our glucose that we can for our nervous system. So this means that we're going to see glucagon in all the opposite times from where we saw insulin. So glucagon is going to get secreted during the post-absorptive state. So post-absorptive, meaning well after you've had a meal while you're just doing your daily business. It's going to be triggered by the same things that would trigger like your fight or flight response, right? So the parasympathetics, rest and digest, are associated with our absorptive state. Means our sympathetics are fight or flight. You're not absorbing things anymore, you're doing stuff. So our sympathetic nervous system and epinephrine secreted by our adrenal medulla in response to the sympathetic nervous system are going to be increased along with glucagon during the post-absorptive state. So during the absorptive state, when insulin starts to take control, the secretion of glucagon is going to decrease. And this is mainly triggered by the increase of glucose in the plasma, which is our main signal that you've eaten something, that we're doing digestion, but we looked at some other indicators that you've recently eaten something. We looked at our GIP being secreted by the small intestine. We looked at small proteins in the blood that can also trigger the release of insulin, but we basically just have these two opposites. Okay. So for looking at a little flow chart, it looks something like this. When, let me move this out of the way. All right, so we're coming from our alpha cells in the pancreas. As we enter the post-absorptive state, we're increasing glucagon secretion from those cells. These are mainly going to have effects on the liver and on adipose tissue, because that's where we have most of our storage molecules stored up. So the liver was where we had a bunch of glycogen. So in the liver, we're going to increase the breakdown of glycogen. Our term for that is glycogenolysis. And so by contrast, we're not going to make glycogen anymore because that would be counterproductive. So we decrease glycogen synthesis. Our liver also is where we can make some new glucose. So we start making glucose in the liver, gluconeogenesis. And we'll also make some ketones at the same time, because we can use ketones for energy, and even your nervous system can use them a tiny bit. Right. We'd also increase protein breakdown, right? We're just breaking stuff down, breaking anything down that we can't use for energy into things that we can use for energy. So decreasing protein synthesis. The only thing we're really making here, the main thing we want to pay attention to, is we can make glucose during this time period. I know it says ketones too, that's true, but less important. So the main thing I want you to think about us making during this time period is really just the glucose. Everything else, think about it as being a breakdown. Okay. Then the adipose tissue, we're breaking down the lipids, which means we're not making triglycerides at this point because we're focused on breaking them down. So blood glucose is under negative feedback control. So anytime we go out of our normal blood glucose range, we're going to expect, hopefully, either insulin or glucagon to respond to bring us back towards our set point, to bring us back in that range that we want to maintain with homeostasis. The normal blood glucose levels are between about 70 and 110 milligrams per deciliter. So anything outside of that range, we're going to have a term for it. So hyperglycemia, too much glucose. That's a problem you might see. So hyperglycemia is above 140 milligrams. Hypoglycemia, also a problem, not enough glucose, below 60 milligrams per deciliter. And either of those are going to cause problems for your body, cause changes in behavior, changes in function, which is why they're under negative feedback control. So insulin and glucagon are the primary ways that we keep blood glucose regulated in the middle there. So as we start to have blood glucose rise, so say we go above 140 or we're getting towards 140, what hormone would you expect to kick in as we have blood glucose rising? Insulin, yeah, exactly. So insulin would bring us down. And if we have blood glucose dropping, what would you expect to click in? All right, I'm going to assume someone whispered glucagon. Glucagon would bring us back up. All right, so we're just keeping ourselves in this normal blood glucose range, ideally. I mean, eventually when you start testing people's glucose, yes. I might set it like, say, normal blood glucose is around 100 or something, because that's an easy number to remember. That's what I pick.
So with diabetics, you want them to keep the blood sugar up more because they can't regulate it.
So diabetics, you want to keep them in the normal range, which is why they take insulin, right? Like after they eat, because they're not producing enough insulin or their tissue isn't sensitive enough to insulin, depending on what kind of diabetes they have. So yeah, you don't want their blood glucose to drop too low, but being too high is also a problem and damaging the cells in the body.
Yeah, I've worked with a bunch of diabetics, and like all of their ranges were like try to keep them within 100.
Yeah, right. Yeah. Yeah,
Even
that
like 200 was like
was good. Yeah. Yeah. If you're diabetic, you can expect your range to like vary more than this because it's a problem with regulating this, right? Yeah, I would say hypoglycemia is more dangerous because, you know, if your nervous cells don't have glucose to function, like that's an emergency. Whereas blood glucose that's too high is bad for you, but it's kind of a longer term negative effect, you know.
What would you say is like a fatal value, like high of like hyperglycemia?
I do not know the answer to that.
Like what would happen? Because I know like hypoglycemia, if you're like too low,
Mhm.
like you can't function, but like is there a point
There probably is. You should look that up and tell me. What I know is like that I think that's part of why like diabetics have difficulty with, you know, like they stop feeling their feet and like various tissues like start to fail progressively. So I don't know if there's like a immediate like this number is too high, if you hit it you die, versus it like damages your systems over time. Are you seeing anything? You Google it
it
now.
looks like over 300?
Okay. Yeah.
So pretty high.
So pretty high. Good.
So that's the result in home.
All right.
So
All right.
I'll take that.
Yeah. All right. 300, everybody. Stay below there. Don't go into a coma. Okay. Yeah. So this is how it's working, basically. So here's the high end as we have that glucose going up. Hopefully we're not going up to 300. Ideally, we're not going past 140, but you know, everybody is different. Everybody can do what it can do. So our beta cells should be increasing insulin secretion in the pancreas. Or if you're diabetic, you might want to take insulin knowing that you've consumed glucose so that you'll have insulin in your system to do these various functions. So in most tissues, this is going to cause that glucose to get sucked out of the blood and into the cells. That's how it lowers that blood glucose level, is an increasing glucose transport into cells. So we're not like totally using up the glucose immediately. First, it has to get into the cells. In the liver and muscle cells specifically, we're going to make glycogen after we've soaked up that glucose, and the liver is going to stop making new glucose because we have readily available glucose coming in from our blood. All of this has the effect of dropping down the glucose in the plasma, which is our negative feedback loop, because we know our negative feedback going up, and we know that if our glucose drops way down, our glucagon would kick in and do the reverse process, which looks like this. So you can trace through these on your own as you're thinking through this, but basically we're just thinking about getting glucose out of the blood with insulin and building out molecules, or saving all our glucose that we can for the nervous system and breaking down storage molecules. So here we have a bunch of tables for you to review as you're studying if you want a quick handy chart of all the various hormones we've talked about with these two chapters. But know them to the extent we've talked about them in lecture. These tables are pretty detailed, or can be pretty detailed. Okay, we're going to get started with our muscles. So first, we are going to be learning the general anatomy of the skeletal muscle, but this is going to be a little different than what we've been doing in lab because we're looking at a more microscopic scale, right? So what we're talking about now is like, we're thinking about like within any named skeletal muscle, we're going to have these additional layers. Right. So first, around the outside of our skeletal muscle, we're going to have the epimysium. So that's surrounding like our named muscle. Then we have perimysium that starts to extend into the muscle. So here's our perimysium. Okay. The perimysium's role is to divide the muscle into these things we call fascicles, which are bundles of muscle fibers. And we say muscle fiber, that's really the same thing as a muscle cell, basically. So we're bundling together many different muscle fibers or muscle cells, typically in groups of like hundreds to thousands. Right, so that would be a fascicle. So here we see they've zoomed in on a fascicle, but this would be another fascicle, right? Like another group of muscle fibers and so on. Okay, so those are all separated by the perimysium, and the whole thing is surrounded by the epimysium. Now, once we're looking at a specific fascicle, we want to zoom in and we want to look at our specific muscle fibers. Right, so surrounding our muscle fibers, we're now going to have endomysium. So endo means in, which is how you want to distinguish it from the epimysium. So the epimysium was like around the whole outside. Now the endomysium is around the muscle fibers themselves. So that's this like slightly darker gray stuff kind of, or blue. I don't know exactly what color that is. Okay. And then we get to the membrane, the plasma membrane of the muscle fiber itself. So now we're actually basically talking about our muscle cell. So the plasma membrane of a muscle cell, of a muscle fiber, has its own special name. It's called the sarcolemma. So the sarcolemma, just our typical membrane bilayer, but it's around a muscle cell specifically. And a muscle fiber, this muscle cell, actually looks different than our normal stereotypical little round cells that we've been talking about. For one thing, they're super long, right? They span the length of the muscle. That's because embryologically, they're basically formed by a bunch of cells kind of merging together. So if you were looking at a muscle cell, you'd also notice that it has several nuclei. So muscle fibers are multinucleated, which is a way that we can distinguish them. That's a way we can tell we're looking at a muscle fiber. Like we have a separate name for the plasma membrane, the sarcolemma, we're also going to have a special name for the cytoplasm here. It's still basically the same concept we've been thinking about. So our sarcoplasm is just the cytoplasm of a muscle cell. So anytime you see this word sarco, you should pretty much immediately know we're talking about muscle, right? Sarco is just this kind of special term that we throw into our muscle words. Right. So when we're looking at the muscle fiber parts, we're going to be focusing most of the time on the actual contraction of the muscle. So we're going to be focusing on our sarcomeres, these structures, which are part of the contractile machinery of the muscle cell. And the larger kind of string of these sarcomeres, which we're going to spend a lot of time talking about, is called the myofibril. Okay, so the myofibril is like these whole long chains of sarcomeres.
So one myofibril is one sarcomere.
One myofibril would be like a chain of sarcomeres and basically.
Okay.
And I'm just like centering this on the sarcomere there because that's the level at which we're going to be discussing muscles mainly. So it's the easiest way to think about the myofibrils is just to realize that we don't have just one sarcomere in a muscle cell, we have a big long chain of them. Okay, so those are like these threads that are going to let the muscle fiber as a whole contract. We're also going to have some other machinery in the cell. The main thing we want to think about is the Sarcoplasmic Reticulum, which is like a modified version of the endoplasmic reticulum in other cells. But its main role in a muscle fiber is to store calcium. We're going to see that calcium is key to how our sarcomeres and therefore our myofibrils and therefore our muscle fibers and muscles as a whole are going to contract, how they're going to get shorter, how they're going to actually allow us to move things by changing how long a muscle is. So when we're zooming in on the sarcoplasmic reticulum, it has this structure we call a triad. So a triad is a tubule, this T tubule. So that's part of the sarcoplasmic reticulum. And then we have these lateral sacs, these kind of swellings on either side that are where the calcium itself is stored. So we have a tubule with calcium stored on either side in these lateral sacs. I could even write in, here's where our calcium is hanging out.
Is it in the blue or is it in the, like, side?
The calcium is in this bluish purple and the green is the T tubule. And so you can see that this is like surrounding basically all these myofibrils. So we're going to be able to get calcium to each of these myofibrils because we're going to see that's how the actin and myosin, which are contractile proteins, are going to interact with each other. We did talk about the neuromuscular junction briefly when we were talking about the somatic nervous system. So here's kind of a plug back to that tiny little 12-unit PowerPoint. So the somatic nervous system is what's going to be triggering our muscle fibers to contract. So if we zoomed in here, where we have our nerve coming in, that's where we would see our neuromuscular junction. So between the nerve and the muscle. So this would be our axon terminal. Here's our motor end plate on the muscle fiber itself. Otherwise, it's mainly working the way our synapses work. We just had some slightly different terminology here. So motor end plate, I think we called it a terminal bouton, but it's the same thing with synapse basically. But here it's going to be triggering these structures in the muscle fiber that we're going to be talking about. So as we're thinking about these myofibrils, these sort of contractile threads, we want to be aware of the fact that they're made of filaments, basically proteins. We have thick filaments and we have thin filaments. The thick ones are made of a protein called myosin. The thin ones are made of a protein called actin. And we're going to be looking at the structure of both of these. So thin filaments, actin, thick filaments, myosin. Come up with any tricks to remember which is which? Let everybody know, because that's key to knowing what we're talking about. So these filaments are in the myofibrils in these long structures. So here we see them here, our thin filaments. They draw for us in blue at the ends of our structures that we're going to see are called sarcomeres. Our thick filaments, they've drawn in this kind of orangey red in the center. So these myofibrils, we're going to think about in these units, these subunits called sarcomeres. But they're all like in a chain, multiple sarcomeres end to end, forming the myofibril. And as you can see, there are several of them within a muscle fiber itself. So we're going to be way zoomed in at this level. But when you start to think about how this affects a muscle as a whole, remember that there are lots, basically. So we're going to think about the structure of a sarcomere, so one of those tiny little building blocks in our myofibril. I'm going to talk about these a little bit out of order because I think a different order to this is a little more helpful to zoom us in. But I like to start thinking about our sarcomere from end point to end point. So our sarcomere is from this squiggly zigzag line, and then it reaches across to the next squiggly zigzag line. So that's the structure of our sarcomere. So this would be one sarcomere. So I like to first then know what those zigzag lines are, right? Z for zigzag. I assume, I don't know if that's officially for zigzag, but I suspect it might be. So our Z lines are where our thin filaments link together. So that's our actin linked together all across kind of a cross section of that myofibril, basically. So Z line to Z line is one set of actin to our next set of actin. The next thing I like to think about is what's in the middle. So what we have in the middle here, M for middle, our M line connects our thick filaments. So this is what I talked about first. The Z line is our thin filaments, which are actin. Our M line in the center, in the middle, is our thick filaments. Conveniently is our myosin, so that's a lot of ends all together. That's kind of helpful. All right. So you can see it's linking together these kind of wheat-like looking structures down their centers. Okay. So that leaves us with the bands and zones. And the bands and the zones are, they exist because before we could look at things on a really teeny tiny level, they had to look at stuff under microscopes. So all they could see was these dark bands of varying levels of gray. So the A band, H zone, and the I band are referring to these kind of histological structures that we can now correspond to understanding how they relate to the actin and the myosin and the varying degrees of overlap. So you can start with the A band. So the A band is like this thick bit of gray to that thick bit of gray. And it's just covering anywhere you see myosin. So it's like the whole thick filament. That's our A band. So anywhere you see a thick filament, whether or not it overlaps with a thin filament. So if there's myosin there, you are in the A band. Within the A band, in the center of it, you can see that there's this lighter part. So this lighter part in the center of the A band is where we have myosin, but no actin. So it's just the thick filament. So that's why it's lighter, because there's no overlap, basically. So that's our H zone. And it almost kind of looks like the center of an H, right? Like if you think about it like that. I don't know, I'm trying to make it work, guys. But H zone in the center of the A band. So myosin with no overlap. Okay. So that takes us to our I band. So our I band is here outside the A band. And so since our A band is anywhere there's myosin or anywhere there's thick filament, are you going to see any myosin in the I band? No. By definition, if there's myosin, it's part of the A band. So the I band has only thin filament. So I band, thin filament, their actin. So if we look at the structure of the sarcomere that we see more often, this cartoon version, it's just this little bit from where the myosin ends, basically to the Z line. The only reason it's circled farther is that it's looking at that piece in the next sarcomere as well. You can also see on the histological slides, you can see that the M line, that connection between the myosin, and the Z line, that connection between the actin, are also visible on the slide. Questions about these lines and bands? Because I know this can seem like a lot of memorization of random letters. Conceptually, see how it like kind of works. We can come back and quiz on these and make sure we got them straight. But that's basically how we're thinking about this. We're thinking about where the myosin is, where it's connected to itself, where the actin is, where it's connected to itself, and then naming these regions where there either is myosin, is actin, or where we have both. So we're going to zoom in on the thin filament on our actin. So actin is one of our contractile proteins. So one of the things that's going to help our muscles contract by helping our sarcomeres contract. And we're going to build it up from smaller subunits. So our first little piece of a thin filament is just one little G-actin globule. Every G-actin has a site for binding myosin. So something sometimes people find tricky is that our myosin binding sites are on actin, and our actin binding sites are on myosin. So try to keep those straight in your mind. It's just because they're trying to connect to each other, right? So the opposite one is going to have a binding site for the opposite molecule. Okay. So these little olive looking things are our G-actin with the site where we're going to bind onto our myosin. Okay. So they're going to form to these long strands that actually form this kind of double helix structure. So if we make one chain, we call this F-actin. I don't know what the F is for, maybe filament. It's good to at least imagine that it stands for filament. Then we have our double helix strand here. So they wind around each other. So then we need to think about our two kind of helper molecules, tropomyosin and troponin. So they're not parts of the actin themselves, but they're forming this kind of complex with the actin that are going to control how the actin works. So the tropomyosin are these long pink strings that we see here. And what do you notice they're overlapping? What part of the actin are they covering up? Exactly. So they're covering up that little dark circle, which is our myosin binding site. So do you think that myosin is going to be able to bind onto actin if we have the site covered up?
No.
No. Yeah. That's the whole point of the tropomyosin, that if we have the tropomyosin covering the myosin binding site on actin, long sentence there, but if you have it covering over this binding site, you can't bind because it's blocked. As we contract, we're going to bind onto the myosin and we're going to slide our filaments past each other, and then we're going to let go. So it's basically while they're contracting, they will have to be able to bind to each other.
Where does this little extra protein go when we contract?
Yeah, exactly. That's the role of the troponin, that little kind of like snowman looking thing in pink. It just pushes it to the side. So it doesn't like get deleted or erased or, pulled off the actin. It just gets pushed off the binding side. So just a little bit to the side. Perfect lead in, perfect segue. So here's our troponin complex. So you can see it's lying right next to the tropomyosin, right? So you can think about it like, like a finger or a peg or a hook, and it just pulls the tropomyosin a little bit to the side. And as it gets displaced, right, if you imagine instead of being here, if our tropomyosin is now here, because we had a molecule push it to the side, that uncovers our myosin binding site and means we'll be able to link together, bind together the actin and the myosin.
That one, like, is that just when we're at rest? Like, unless we're at rest?
Yeah, basically, well, we're not contracting, yeah.
Okay.
Yep. And we're going to see that there's this whole cycle of myosin and actin binding together, the Crossbridge Cycle, that lets us pull past each other. So the troponin, this little snowman thing, technically has 3 proteins that are part of it. That's why it's these 3 things stacked on each other. So the I component is binding to the actin. The T component binds to the tropomyosin. So T for tropomyosin, don't have a trick for the I. But the C part binds to calcium. So that's a reversible reaction. So when calcium binds to troponin, that's basically going to activate our troponin and tell us to uncover these myosin binding sites. And that's how we're regulating the skeletal muscle contraction. So we'll have calcium bind in, and calcium can also remove itself. So when there's no calcium anymore, we're going to cover those sites back over with the tropomyosin. So that's the way in which calcium is going to sort of trigger the skeletal muscle contraction, is by allowing us to expose this binding site. So now we're going to look at the thick filament. We've just been looking at the actin, the thin filament. We know that it has a binding site to bind onto the myosin. So now we're looking at the myosin. So when we're thinking about our sarcomere, and I'm just going to kind of draw one at the top, right? So here was our actin on either end. Those are our thin filaments. Here are our thick filaments, the myosin, kind of like that. I should draw a little bit like that. So this is the general structure. And I'm just drawing it this way because I've kind of only shown you half of a myosin molecule for some of these. So if we were to add on to it, you look closely at the cartoon pictures, it has like these little lumps coming off the ends. So that's what we're zooming on here in this A part. We've zoomed in on like this part of a thick filament, so that part of a myosin molecule. So we consider the tail of the myosin molecule to be towards the M line. So here would be our M line. So we basically have like two golf clubs sitting end to end, like kind of handle to handle. And that M line is where they meet. Okay. So that's the tail. And the heads are towards the I band, so kind of towards the Z line, towards our actin. On each head, we're going to have a place to bind actin so that we can move these two types of filaments against each other. And we're going to have a site where we're actually going to break down ATP, because we're going to need energy in order to contract our muscles, right? Like you need to expend energy, you need ATP in order to do this. Actually, the way ATP works, right? The way ATP actually gets used for energy is you break it apart, right? It's fundamentally the way ATP works is it's that breaking of a bond in that third phosphate group that releases the energy for other molecules to work. So here on the tips of the myosin heads, we see our actin binding site. So that's the part that's going to bind onto the corresponding myosin binding sites on the thin filaments that, when we're at rest, are covered by the tropomyosin. Okay. So, yeah, and here is our binding site for ATP. So that's in purple. So we're going to need to be using both of those as we slide these filaments past each other. We also sometimes refer to these heads as our cross bridges. We're going to see that we refer to our whole cycle of moving these filaments past each other as the Crossbridge Cycle, because we're going to see that we basically have kind of a movement in the position of these heads. Like we're going to have them sometimes in this position, and then sometimes they're going to paddle down like oars into that position, and that's how we create the movement, basically, is just by a change in the position of the head. Okay. So realistically, when we're looking at the whole thick filament, we have a lot of these together. So our M line would be in the center of like what we call the bare zone. And then we'd have a lot of these myosin molecules, tail to tail, with their heads at the end, all kind of wrapped and clustered together. So we have these happening all at different levels here in this kind of section where we have a lot of cross bridges. So we're going to be focusing on like what one head or one myosin molecule does as we talk about the Crossbridge cycle. But again, if we zoom out our mental picture, we want to be aware that this is happening in a lot of different places. The final little thing I want to see here, we have this one other molecule involved here, an elastic protein called titin. Titin is actually the only thing that's really going to seem to kind of change length here in terms of a protein that changes length. We're going to see that while our sarcomeres shrink, it's actually because we're sliding things past each other. The molecules are staying the same length. But titin is a spring, right? It's a coiled structure here, basically to just stop those thick filaments, that myosin, from like sliding way out of location, right? So this is a supportive protein, just kind of anchoring those thick filaments. So we're not going to really see it or think about it again, but it's helpful to realize by contrasting them with titin that the myosin and the actin don't change length. It's really only this titin. And the titin isn't what's making the movement happen.
What is the human data that doesn't look like it?
That's a good question. The level to which I know that is just the connection between the actin. I don't know if it's a specifically different named molecule or whether it's like a different form of actin. I'm going to briefly introduce fourth generation, but we'll really talk through this cycle when we meet on Friday. Okay, so but the basics of the fourth generation, the basics of how a muscle contracts, we have something called the sliding filament model, which means like, this would be easier if I could use my mouse to hold this pen, whatever. Okay, so this pen is the thick filament of myosin. My hand here is the Z line, the actin. So the way sarcomeres shorten is just, we're gonna have that actin slide in like this. That's it, that's all that's happening. That's the sliding filament model. So nothing is actually really shortening in terms of molecules. So we're gonna see some of those bands that we looked at on the histological side shorten, because those bands were naming where we had myosin, where we had actin, and where we had overlap. So the amount of overlap between these two molecules is changing, but the molecules themselves are not changing at all. So that's the sliding filament model. That's how a sarcomere gets shorter. And because our myofibrils are just sarcomeres end to end, and our muscle fibers as a whole are full of myofibrils, that's how a muscle itself contracts, which pulls its two endpoints closer together. The endpoints are usually bones, so the bones move closer together, and that produces movement. Now, from a physiological level, we want to understand how we're generating the force to do this. Like, where are we getting the energy to move these molecules past each other? How are we actually making them do that? So that's when we're going to zoom in on the Crossbridge cycle. It's going to be talking about how those heads of the myosin, how they change position, and how that lets us move these two types of filaments past each other. After we talk about the Crossbridge cycle, which is where we'll spend most of our time, we'll talk about how we turn that on and off using the calcium and using signals from our nerves. So that's going to be the excitation contraction coupling. And then eventually we'll move into muscle cell metabolism and talk more in depth about ATP and how that drives the cycle. Okay, so that's our task for Friday. We're going to stop here for today.